Hey everyone, thanks for tuning in. All right, so over last summer and well into the semester, Dr. Pringle and I, we worked on this project. So here goes. So our objective was to identify different species of microbes living in different sediments of Missouri, Illinois, and Mississippi rivers. They were all relatively close to each other. We used really new nanopore sequencing technology, which was the first time was going on in Maryville. And we also wanted to identify antimicrobial resistance indicators within the genomes of these species. I put a link down here. I'm not sure if the mouse shows, but I put a link down here for anyone who wants to learn more about how nanopore sequencing works, because it's very different from how traditional sequencing techniques work. And then this is important because knowing what's going on in the microbiomes around us, knowing what's going on in river sediments, it can tell us a lot about what we're doing to the environment. It can tell us about what's going on naturally within the environment, and it can also indicate what's going on within bacterial species that may be acquiring resistance to drugs we're taking, or maybe even acquiring resistance naturally to drugs that we're using clinically. Please feel free to let me know if my voice isn't loud enough at any point. Thank you. So this map here shows us where we collected our samples from. So we first went on to the Missouri River, the Mississippi, and then the Illinois. This was all on a hot summer day during the morning. And whenever we collected our samples, we were very careful to use gloves and avoid contamination. As soon as we collected the samples, we went on and stored them in ice to make sure everything was as less contaminated as possible. And as you can see, the rivers are not really that far away from each other. So there's not like any drastic factor of climate or anything that could affect our results. So here's, this is a general overview on our DNA purification part of the methods. The first couple of steps involve cell lysis, which just means breaking down the cell membrane, the cell walls, trying to get into the part of the cell with the DNA in it. And then the rest of the steps, it's DNA or RNA. And then the next couple of steps are purification steps. They are steps where we try to re-extract the genetic material so we know it's really clean, and then just storing it. After we have our genetic material, we went ahead and prepared our genomic library, which included ligating our DNA sequences because most of it may have been chopped up, and attaching adapter sequences so the sequencer is able to identify different strands. And I want to point your attention to this little chart here. So whenever we ran our sequencer, what actually happened was I talked about nanopore sequencing, and here we have a nanopore, a small pore in the minion, which every time you have a different DNA base, there's a change in voltage. Now, based on this change in voltage, we can use applications such as Guppy right here. And here's just a little screenshot of the command line interface we had to run to get all our bases identified. And once we had all our bases, we were able to run it through an application called Epi2Me, which was provided by the manufacturers of the Minion sequencer. And what this does is it tells us which reads mean or where they come from, what species, and all that. And then after having our data from Epitome, we were able to classify it using, or not classify, we were able to sort it through our studio because it was a lot of data which we couldn't handle individually. We had to use some other kind of program to kind of break it down for us. So here's a general overview on the results of the 3 rivers, and I included a little picture of where we went and collected the samples from. They're all from right by the riverbank. And as you can see, we have a ton of bacteria in every sample. We have very, very low amounts of viruses or archaea, but I think the fact that we're still picking up on the viruses, archaea, and eukaryotes, I think that's really cool. But we can see that we have a lot of bacteria and relatively low amounts of all the others. So when it came down to analyzing the data we sorted using RStudio, we decided to go ahead and look at the top 15 genera relative to the sample in every sample. It makes it a lot easier to really understand what's going on when you have 15 different colored boxes versus 25 or any larger number. So all these tables here, they're all color coded, so you can tell using the key what box means what. And you can see that in every river sample, the top 3 or top 4 tend to be shared. We have Burkholderia here, Pseudomonas, Streptomyces, and Bradyrhizosium in all of them. And also you can see some Homo DNA here. We can also see some Homo DNA here as well. Now, what does all of this really mean? So I created this Venn diagram over here to kind of show where different genera overlap. And we can see that a lot of them are really condensed in the center. And after doing some research on these species or genera that we're seeing, I found out that they are all aerobes and they all were recorded to live in soil and water. And then we see on our odd groups, we have a plant pathogen over here, a common plant pathogen. So this could tell us something about plants living in the Illinois River. And then pointing your attention down here to the Mississippi River portion of the outgroups, there is Ribosium, which is a nitrogen-fixing endosymbiotic bacteria, which could tell us about the plants it's forming associations with. Maybe those are present by the Mississippi River, but not here or in the Illinois River. So also the fact that we were able to find some human sequences in the sediments, this can indicate the presence of treated or untreated human waste being discharged somewhere close by the rivers. And Dr. Pringle actually mentioned to me that there are 2 separate sewage treatment sites right upstream the Missouri River, so this could be very possible. And then, yeah, we can see that some genera were found predominantly in some river samples. This tells us that local conditions possibly affect the presence of microbes, or not possibly, they definitely affect the presence of microbes, because depending on your environment, you're more likely to survive somewhere than somewhere else. And like I mentioned before, in addition to wanting to find out what species we were finding in the different rivers, we were also interested in finding out about the antibiotics they're resistant to. And just like with the whole species thing, we decided to look at the top 6 antimicrobial resistance markers instead of every single one we identified for purposes of simplification. So here we have the microbe, we have the river up here, and then the drug the microbe is resistant to. Again, this is color-coded, so it's easy to follow on what marker is the most popular, how many variants we have of it. So pointing your attention to here, RE is the gene that is mutated within this Pseudomonas aeruginosa strain. It is resistant to fluoroquinolones, Mycobacterium tuberculosis. Like the name suggests, it does cause tuberculosis, and we are finding it in our waters. I think this is concerning. And then we have Salmonella over here. And you can see that we have resistance to fluoroquinolones and isoniazid in every sample. So what is the significance of these antimicrobial resistance markers? So a few of them, or several actually, they all are of clinical significance. So isoniazid and rifamycin, like we see here, which come from Mycobacterium tuberculosis and tuberculosis as well. They are both used as first-line treatments for tuberculosis. So when someone gets tuberculosis or is diagnosed with tuberculosis, these are one of the first few medications that are administered to them. And then two antibiotics in the fluoroquinolone groups that we saw. They're used to treat Pseudomonas aeruginosa infections, who are ciprofloxacin and levofloxacin. Spectinomycin is used to treat respiratory and enteric infections in animals. And then inhibitor GE2270A is active against gram-positive bacteria, including MRSA, which is known to cause a lot of infections or opportunistic infections in hospitals once someone is already infected with something. Other findings, other than markers being of clinical significance, we see that Streptomyces species, they produce alphamycin, so we expect resistance from them, even though it's not in a clinical sense. Pyromycin is active against prokaryotic and eukaryotic cells. It inhibits their protein synthesis, so they can't really make anything for survival. But this is used in the lab a lot. So to conclude, the predominant bacteria in each of the river samples were very similar. This shows us that the environmental conditions were affecting the microbiome more than the water composition. Some of the bacteria detected possess genes that confer antibiotic resistance. So like we just saw, we saw bacteria that were resistant to clinically relevant medication. And also further analysis is going to be required to identify whether these antimicrobial resistance markers are intrinsic or acquired, meaning is this something they got or is this something they had in them? Again, back to the Homo DNA we found, 2 sewage treatment plants were identified upstream of the sampling site, so very likely that this is just a result of untreated waste. For follow-up work, we would really like to investigate antimicrobial resistance markers before and after sewage treatment. What antibiotic resistance markers are present in human sewage before treatment? I'm very excited for that portion. I think it will tell us a lot about the kind of medication people are taking, whether there's a selection against it, a selection for it. It can also tell us whether we need to do anything about the medicine we're taking. Are they effective? We also want to determine consistency of different microbial genera at different parts of the river. That is something I wish we had done along with this project, honestly, for the sake of consistency with the results or accuracy. And then we want to determine different microbial composition at different seasons, or during different seasons. So like I said, we did this in a hot summer morning. So the results could have very easily been different if it were during the winter, during the fall, or anything. Here are my references. And thank you. Feel free to shoot any questions.
And feel free to check the chat for questions, Fudge. And
Oh.
This is Rebecca Dorman. I have a quick question. Can you hear me okay?
Yeah, I can.
Okay, perfect. Early in the presentation, you mentioned that one of the techniques you were using, or the machine, was one of the first times that Maryville has used that in student research. And I wondered if you could share a little bit more about like how you feel it might be using it in the future, or like where do you see it going from here, even at Maryville?
Yeah, for sure. I already know a ton of students who are interested in using the Minion. I think it's not very hard to use at all. It's very straightforward. It does take a lot of practice. I was very nervous using it at first because it was this expensive piece of equipment. And I had to assume that what I was doing wouldn't mess it up, which I was really careful, so it didn't. But I was really excited to use it. And I know that in the future, a lot of people are going to come up with really, really cool ways to utilize it. I already know a couple of things that some people are doing or that they want to do. And I think it's really cool.
Awesome. Thank you so much. Congratulations on a great project.
I'm not sure I can look at the chat from here, so I'm just going to stop sharing for a second. Oh, you're muted, Dr. Kobeck.
the first question you got was from Dr. Ulrich. She said, do you know how to use R now?
Yes, I know how to use R now.
So you're going to be in high demand. And
that's exactly right, yeah.
I asked the question, could these resistance genes survive the water treatment process and make their way back into our water supply?
Very possibly. I don't see a reason why not.
So we're treating microbes, but we're not treating DNA.
That is true. That is true.
And then Dr. Ulrich also asks, I wonder how water treatment plants are testing for that. Do you know if they have the capability of testing for the presence of nucleic acids in water?
I'm not sure if they have the ability to test for nucleic acids, but I feel like that's not really something they're doing. I know that one treatment plant somewhere by St. Charles, I know they don't look for nucleic acids because we were in contact with them about us doing it. So I know they don't do it, but I'm not sure about every other treatment plant.
And then in theory, it would be possible for those resistance genes to get transformed into living bacterial cells, right?
It is, yeah. But if you're breaking down the nucleic acids using ultraviolet radiation or anything,
yeah.
I'm not sure we're gonna have to see the next project.
Okay. All right. Well, thank you very much. Excellent talk. Appreciate your contributions. Thank you, Fudge.
Oh, I just want a question. Any drawbacks to using the Minion? So, Mike, so far I don't see any drawbacks using the Minion except for the part where you have to wash the cell after every use.